Hello, everyone, and welcome to the third episode of the Salivary Gland Research Podcast. My name is Joey, and it is my pleasure to introduce the participants of this week's episode. Serving as moderator, we have Dr. Sarah Pringle, who will be interviewing Dr. Catherine Ovitt, who has done great work in the area of salivary gland regeneration during her time at the University of Rochester. Now, I will hand it over to Dr. Pringle, who can start with some introductions.
Thank you, Joey. Hi, everybody. My name is Sarah Pringle. I'm an assistant professor at the University of Groningen in the Netherlands, and I work in the salivary gland field, beginning originally about 15 years ago with irradiation damage within the salivary gland world, and more recently moving into that within Sjogren's disease, we now call it. Yep, so a relatively established career within the salivary gland world, but I know that Dr. Ovitt has a much more established career in this field than I have, so it is my pleasure to introduce you. To start our podcast, maybe it would be handy if you could tell us something about yourself and how you got to where you are.
Sure. Thank you. Thanks for the invitation to be interviewed on this podcast. It's quite exciting. So I'm Catherine Ovitt. I've just retired 10 days ago from the University of Rochester, where I have been working since 2001. And prior to that, I actually I grew up in the military. My father was a sergeant in the Army, and I went to the University of Connecticut way back in the 70s and got my bachelor's degree there. Because neither of my parents had ever been to college and I really had no idea how science worked, but I loved biology. I ended up taking a job as a technician straight out of college, and I had to pay a lot of school loans back because there were 7 kids, and so my father didn't pay for college. And so I worked as a tech for 4 years. During that time, I realized that it was kind of a job that could be a little frustrating. And I decided after a couple of years to try a graduate course or two. And I, at that point, was at Temple University in Philadelphia as a technician. And I was encouraged eventually, after 4 years, to apply to go to grad school. So I ended up in St. Louis, Missouri, where I went to Washington University, and I got my PhD in molecular biology. At that point, I had always been saddled with a lot of school loans, debt, and very little chance to do anything. And I realized that one really great thing one could do in science is get out of your normal sphere. And I was encouraged to look around outside the United States for a postdoc. So I ended up applying and being accepted to be a postdoc at the European Molecular Biology Lab in Heidelberg, Germany, which was a fantastic place to work and a really excellent experience for any American, I think, to get outside of this continent and get a different and much broader view of the whole world. And certainly at the EMBL, you do, because the EMBL is a consortium of scientists who come from all kinds of countries, mostly from European countries, but others as well. And the language that is spoken there officially is English, but of course you're in the middle of Germany, and you're working with scientists from almost everywhere, and certainly from different parts of Europe. And so I ended up staying for a second postdoc after my first 3 years, and then I met my husband, and so I stayed for a third postdoc. I ended up kind of running out my time at EMBL because they have what they call a 9-year rule. People have to leave after that point and move on and take your science and take your experience and go back to your home country. That's the general idea. But at that point, I was married and I had 2 kids. So I left the bench and I went to work for Springer. And Springer is, the publishing company.
Sure.
They do a lot of scientific journals. And I was a copy editor at Springer for 4 years until my husband was recruited to the University of Rochester. And at that point, I'd been in Germany for 14 years. I didn't really want to come back to the States, but we came to Rochester. And part of the draw was that Larry Tabak was running the Center for Oral Biology um, in Rochester at that time. And this was prior to him moving to NIH. Um, I had been the whole time at EMBL working on embryonic stem cells and learning how to make chimeric mice, doing a lot with transgenics and chimeras and knockouts. And so I was recruited by Larry to join his lab and bring mouse genetics into this field of oral biology, specifically salivary gland. But unfortunately, before I had a chance to actually arrive back in the States and in Rochester, Larry moved to NIH. So we came to Rochester anyway, and I ended up in Jim Melvin's lab. And after about 4 or 5 years as a research assistant professor, he encouraged me to write a grant, which was funded. It was an R21. And then I was encouraged to apply for a tenure track position. So I came to this whole business kind of roundabout. I hadn't done any oral biology before I came to Rochester. And also I obtained my first tenure track position at an advanced stage. But actually it has worked out really well for me. And so now I've just retired from being full tenured professor. And
Okay.
sorry, that was a long-winded answer, but
That was a very complete answer. You've had maybe a more unusual, if you can say that, route to where you are or where you have just been. I'm not sure whether I should congratulate you or commiserate you with having retired just 10 days ago, depending on how that ended up for you. I didn't know we had a common background in embryonic stem cells. I did my PhD in embryonic stem cell biology as well. But that's aside from the point from today's podcast, otherwise I'll get distracted. I've known you for the time since I've been in the salivary gland world. And if I think about your work, generally this morning I was thinking it tends to fall into, if I don't think about it too much, so that's what you're supposed to do, right? Just think quick off the top of your head what your work reminds me of. Generally, I think about plasticity, acinar cells, self-duplication of acinar cells within the salivary gland, and the ASCL3 stories, which were around towards the beginning of my postdoc times, I think. And I remember very well when your group presented the acinar cell duplication story for the first time at a particular conference we were both at, because there was a very strong statement which sent some kind of shock ripples through the audience and I wrote it down in preparation for this conference, this podcast. Contribution of stem cells to the salivary gland maintenance is minimal, which at the time was, and not so much now at all, actually, at the time was a statement which some of us were perhaps not ready to hear, and in some contexts, maybe also a little bit wobbly still, but it was a very controversial data presentation at the time, but it still holds up. I would like to invite you, if I may, to kind of from these three topics, so your work with plasticity in relation to acinar cells after injury, acinar cell duplication, or the slightly older ASCL3 story, pick your favorite, and where did the inspiration from that course, that work pathway come from?
Well, basically, I started in the same vein and with the same dogma in the salivary gland field that I think you did, which is, okay, there are stem cells there. There are stem cells there, and we've got to find them.
Right, we were all a little bit kind of tainted by the hematopoietic system, I think, at the time when it started with this tree situation. I'm using my hands as a podcast, it's not useful, but this tree situation where you can see cells differentiating from each other, and we had to move away.
Everyone, me included, believed that, okay, this hypothesis that intercalated ducts, sit right at the cusp between acini and ductal cells, and that would be the perfect location for the stem cells. There was, evidence that this could be the case. And so we were pretty convinced, which is, it kind of is interesting because that's where the ASCL3 story came from. I decided, okay, let's try to find some gene that's expressed in the ducts or in intercalated ducts that we could actually mark and look at. It's a transcription factor. It
Hmm.
had just been published in 2001 that it's salivary gland specific, which isn't true, but that's, it looked good at the time. And it is not expressed in acinar cells. So I thought, aha, this is going to be it. So
Yeah.
the first thing we did was to knock in a Cree GFP into the ASCL3 locus. And at the beginning, it really looked very positive in terms of a potential stem cell, because what would happen is the gene itself, transcription of that transcription factor, is turned on quite early in development. I mean, not right at the beginning of salivary gland development, but definitely,
Uh,
prenatally.
oh,
So
okay. So we're talking embryonic still. Okay.
yeah, embryonic. And so if you use that Cree as a lineage tracer, so the Cree mouse, the ASCL3 Cree mouse, is crossed with another mouse that has a reporter. In those days, we just used beta-galactosidase.
Yeah.
So if you cross them, then whenever that Cree is activated, and it's activated whenever ASCL3 comes on, then you're going to get those cells will get the beta galactosidase gene turned on, because that's how it works with the Cree and the activation. And we kept seeing, wow, we have acinar cells that are blue, we have duct cells that are blue, so wow, this must be a stem cell, because these
Yeah.
this is happening early on. And it took us a number of years before we were finally ready to say, hey, wait a minute, this is an embryonic activation. This is during the time before final differentiation of certainly of the duct cells and probably the acinar cells as well. And probably it's just a constitutive Cree that's just going to keep, it comes
Hmm.
on, it keeps making more beta galactosidase positive cells. And so we kind of gave up on the idea that ASCL3 marks stem cells, which brought us to the second step, which was, okay, what does mark stem cells? And in the meantime, a lot of labs had been looking
Yeah.
at that, a lot of labs. And
Hmm.
can't, I don't even want to try naming names because I know I will leave someone out. But there were lots and lots of stem cell so-called markers out there, and we were kind of frustrated. Okay, how do you prove this? And then by going into the literature and looking in a different field. So we started looking at pancreas. And in the pancreas, there was quite a bit of controversy as well about whether or not there's a stem cell um, for the pancreatic beta cells and the pancreatic acinar cells. And so Doug Melton's lab had actually done what I consider a pretty classic experiment. And what we decided to do was to replicate that in salivary glands. And it's very simple. So first of all, we found a gene that's expressed in all acinar cells, but not in duct cells. And that one is this BHLH15, which is MIST1.
Missed one,
Okay.
right?
MIST1 is a gene that is transcription factor that's expressed pretty exclusively in the acinar cells of the salivary gland. And then if you use that gene to drive a Cree, again, Cree recombinase, and cross it again with a marker line. So again, we started with beta galactosidase.
Hmm.
Um, you find that as soon as the MIST1 promoter comes on in the differentiated acinar cells, all of your acinar cells should be blue. They should be beta galactosidase positive. Okay. And so the idea is, okay, what happens after a chase? All you've labeled with your beta galactosidase, your Cree beta galactosidase has been acinar cells. You shouldn't have labeled any duct cells,
Hmm.
and you shouldn't have labeled any of these potential stem cells.
Hmm.
And so if you just follow the chase of this thing, of all of these blue acinar cells, if there's a stem cell that's not labeled, eventually you should start to see replacement of the labeled cells with unlabeled ones that are being generated by the stem cell. So
Right.
basically, this whole experiment was to ask the question, are there stem cells or not?
Right.
And
And it
um,
kind of, sorry to kind of
no,
if we can kind of back and forth whilst I think of things, because I'm making millions of notes on the side here. Otherwise, I will forget my
Okay.
my points. When you think about those kind of experiments, which are really classic, you're basing it on the assumption that you capture the turnover time of the salivary gland within the pulse chase experiment that you run, right? So
Right.
a mouse is fairly fast. What was the
Mhm.
state is it 30 days or 60
It's
days that the
30 to
mouse,
60. We were never really able to pin that down exactly,
right?
but 30 to 60. Yeah.
So you need to be able to be confident of your window of capturing everything.
Absolutely. And we weren't completely. And so, but we did find that the overall percentage of beta galactosidase positive cells didn't significantly decrease
No.
over a 30 to 60 day period. And so that leads you to speculate, okay, wait a minute. Are these cells just dividing themselves?
Yeah.
And so, to answer that question, we did another more complicated genetic trick, which was to, instead of using beta galactosidase as our marker, we used this rainbow or brainbow mouse, which actually it has four different markers. These are all fluorescent markers, and there's GFP, RFP, YFP, and I think the cyan in there as well.
Yeah.
And they're all in a long, like concatemer kind of organization. And any one of those four markers can be activated when the Cree it causes recombination. And the idea was, okay, do a very brief pulse, label a single acinar cell. Doesn't matter which color it is, but you label it, and then you follow it. You allow the mouse to just continue, for 30 to 60 days. And then you look at what you have. And what we had were small clones of individual colored cells, suggesting again, or kind of proving that if you labeled one and then you waited over time, that one divided. And because
Mhm.
it's yellow or it's green or it's blue, it makes a small clone of itself. And it was that set of experiments that actually convinced us that the acinar cells themselves are dividing.
Mhm. I remember that was in science is funny because you can date things by the techniques and everything that everybody was using in those eras, and those were very much in the burgeoning rainbow mouse
Right.
days, which was a beautiful, beautiful model to look at these kind of things. I remember the microscopy
Mhm.
pictures very well.
Mhm.
Let me think. So and of course that's in a mouse model situation, which is fairly easy to manipulate mice and turn them into different colors. I was thinking about the translation of this into the human context, which I know you also approached. So the major salivary glands, and if they are also turnover-wise, time, acinar cells. Is it comparable, do you think, the level of contribution of proliferating acinar cells to maintenance of the adult human major salivary glands to those of the mouse?
In terms of time or in terms of mechanism? In terms of,
Yep. Let me
I
define that
think
better. I
I
think
would I would hazard the guess that yes, it's comparable. I would say
comparable.
yes. I think that this is it's a mechanism, and it's not just the salivary glands. I think there are a number of other tissues
Hmm.
that have similar mechanisms for just basic turnover. I
Hmm.
mean, I'm not talking response to injury, or stress, but basic turnover. I do think that the ability to just maintain, and if you have a slow turnover time, 30 to 60 days, and you have a certain small percentage of your cells that are just, every once in a while proliferating. It's a really important question now, I think, to understand, is it all cells? Do they all have the same
Yeah.
equal capacity? Or is there a subpopulation,
It was interesting for me I was reading your work to look at if I remember correctly, that the parotid gland, human parotid gland, has a slightly lower level of intrinsic proliferation of the acinar cells compared to the submandibular gland. And in terms of how that might affect, for example, I work with Sjogren's disease and then almost exclusively with the parotid gland and the acinar cells within it. And we have a fairly, it's not very defined field in terms of the nature of the acinar cell dysfunction sometimes within Sjogren's disease. So it's very interesting for me to make the comparison. Um, so you convinced us with those series of papers about the intrinsic proliferation potential of the acinar cells. And then, if I remember correctly, there was a series of papers moving on to look at plasticity of acinar cells in response to, so that first package of work, if you like, was very much in a maintenance homeostasis situation, and then moving on to see logically what happens if you provoke the acinar cells,
Right.
basically.
Right.
And for the audience who have not stalked you as much as I have, maybe you could outline that
Yeah.
package of work.
I mean, obviously, we're all focused on, how do you repopulate or regenerate the salivary glands after radiation damage? That's clear. And what's always been a kind of an important and perplexing question is we have this injury model in salivary glands, which, I just learned from other people in the field when I came in, called duct ligation. And if you, what happens is, particularly, it's generally done on submandibular glands because that's the easiest, but it has been also been done on parotid gland. And if you obstruct the duct, the excretory duct that leads to the oral cavity, the gland itself will start to degenerate. What happens is you lose the acinar cells. And so the interesting thing about that injury model is that if you release that obstruction, the gland will somehow regenerate. And unless you leave the obstruction for 2 to 3 months, the gland pretty much can regenerate or repopulate the acinar cell compartment. So the question is, what's the difference? There's a lot of work going into now exactly why acinar cells are so sensitive to radiation that they aren't able to do this regenerative thing. And I know our lab tried to do a little bit of that, but I think there are other labs that are making bigger headway, and I think that's great. But what we were trying to do was to understand, okay, where do the cells come from after this duct ligation injury? Because they're able to repopulate the gland. And so we looked for we did the whole lineage tracing thing again. If you label your acinar cells and you do your duct ligation, you lose all your labeled acinar cells after the duct ligation. The truth is you don't lose them all. You lose a lot of them, but there are still some that remain. They just don't look like acinar cells anymore. And the strange thing about them is they start expressing what we would consider duct cell markers.
Like keratins and that kind of thing.
Exactly. And so that's where we got into the plasticity thing, where you say, wow, these cells are somehow losing their phenotype. They're maybe, I mean, we'd like to think they're reverting to a more undifferentiated state. And so that's where those papers came from. The bottom line there was that we didn't expect to find that the duct cells could do it too, and the duct cells did it too, and they also lose their phenotype, and then some of them end up expressing aquaporin 5 after you regenerate the gland, and that just was so confusing. I think again there are a lot of questions that could be answered maybe by doing single cell sequencing or something like this of these different populations. It's got to be kind of.
It was on the tip of my tongue, basically. Just like these days, we would probably throw some single cell sequencing at it and want to know exactly what the cell types were doing and make a whole trajectory analysis and how things changed between different time points. My question was going to be, did you ever have any kind of idea why some of the acinar cells, like after duct ligation, is it just like a random chance that some populations stayed there and then turned back into ductal type cells? Or was there any logic behind it? Or was that just a silly question?
Not that we could determine with the early experiments that we did. Marit did them. And they were done with RFP, so red fluorescent protein. You could really pretty clearly see the cells that were left. They just looked like blobs.
Yeah.
They were not, they didn't look polarized. They clearly had lost their polarization. Again, there is, there are precedents for that in the pancreatic field and in the gastric,
Mm.
the stomach lining. Cells lose polarity if they're under stress, and particularly secretory cells, and they downregulate a lot of the things, including MIST1, that they normally would be used to make, or would make them into secretory cells. So I think it is a general phenomenon. Um, what we were trying to get at was whether or not one could harness that
Mhm.
phenomenon, but that's a really much more long-term question.
Yeah, I find it fascinating if we think about, because the salivary gland is a relatively, for most people, inconspicuous organ that maybe they don't think about it so often as we do. But it reacts fairly violently to a lot of different stimuli. So radiation, um,
Your
drug
cancer
use,
drugs,
medication
right?
use, ICI, immune checkpoint inhibitor use. It's heavily involved in salivary glands, has been described in salivary glands in Sjogren's disease, and has been described as a viral sink, a collection of viral, stimuli from the outside environment, because it's so close to everything that the carrier of the salivary gland is exposed to. It's a very reactive organ, which is maybe a bit underestimated, in my
Mhm.
opinion. That
Yep.
work was fascinating for me because it mirrors a little bit some later work now about the checkpoint inhibitor use and effect on that little niche between the acinar cells and the intercalated duct cells, and perhaps also the myoepithelial cell triangle and their plasticity in between the three to
Mhm.
apparently smush together into one kind of less defined cell population instead of staying in their defined lineages.
Right. Right.
We are at 30 minutes, more or
Mhm.
less, or so, which without me even paying attention very much to the clock, I was getting carried away. I have a whole load of other questions for you.
Mhm.
Um, Maybe I could round up before we finish this podcast with asking you a less science-based and more journey-based question for you had a very an inspiring path towards your career trajectory. Um, would you have done any of it differently in the way that it panned out?
No, I mean, it's hard to say. I think I've had a great run here at the University of Rochester. I didn't want to come back to the States. I would prefer to have stayed in Germany, but actually, I think within the German academic system, I probably wouldn't have had the opportunities I had here at the University of Rochester. So I think in that sense, um, everything worked out. And I've always said to young people coming into my lab, um, you may not think that salivary glands are interesting, but once you do two or three experiments, you're going to find almost anything you do interesting if you like science, you know?
Right.
Yeah.
And you just start seeing acinar cells everywhere.
Right. But there are
Thank,
other things, if someone threw me into a muscle lab, I probably would have loved
right,
working on, muscle
right.
development.
Or if you'd stayed in embryonic things
Exactly.
more purely to your PhD roots, same story for me. Who knows what would have happened in the end?
Yeah. Yeah.
So I think looking at the time and the clock, I would like to thank you for your discussion today about your career and your research contributions, which have left a very big mark on this of the salivary gland regenerative field. Um, Enjoy being retired, I would
Yes,
say,
I am. I'm
and
playing
not reading
a
any more
saxophone.
papers or grants or anything. And I hope to
Actually,
see
I'm
you
on
again.
study section next month. So
So not completely retired.
no, they don't let you go so easily.
And I hope to see you in the near future at a conference somewhere.
Yep. Great.
Have a nice
Okay. It's
day.
great talking to you. Thank you all.
Bye bye.
Bye.
This concludes episode 3 of the Salivary Gland Research Podcast. I would like to thank Dr. Pringle and Dr. Ovitt for taking time out of their busy schedules to join us in discussion today. I would also like to thank the audience listening to this week's episode. Please make sure to like and leave us with a comment if you enjoyed the episode. We hope to see you in the near future for the upcoming new episodes. Thank you.